Hello, welcome to this week's videos lecture. I'm Dr. Harris-Jennings, and today we're going to be talking about the introduction of microbial metabolism, chemical reactions, and enzymes. We'll talk about how molecules are built up and how they're broken down. We'll talk about how enzymes speed up chemical reactions so that they can happen on the time scale of life. We can talk about how ATP is the currency of the cell and facilitate these enzymatic reactions. And we'll talk about how we generate ATP through the process of aerobic respiration with oxygen, anaerobic respiration without oxygen, anaerobic, the AN is without, so without oxygen, and fermentation. And we'll talk about the electron transport chain. With that, we can get started. So metabolism is the connection of chemical reactions that work together in concert. So chemical reactions, you have a substrate that turns into a product, and it goes through a chemical reaction to get there. And so metabolism is pathways of these chemical reactions or series of chemical reactions where one substrate gets turned into a product, and that product is the substrate for another reaction to turn into a different product. And that goes into another one, and so on and so forth. And this is how we can break down molecules, build up molecules to make what we need when we need them in our cell, or in this case, in our microbes, in our bacteria, fungus, and things like that. So we have many different pathways that are all kind of working together. It's a network of these metabolomic pathways. And so there are catabolic reactions and catabolic pathways that break down material. And usually we try to harvest energy from those when we're talking about metabolism. And when we have anabolic pathways, we're building up molecules. And when we're talking about metabolism, we're usually going to have to input energy into making these molecules to overcome energetic barriers to make them. And so while we're deconstructing and building up molecules, usually we have two separate pathways that work kind of in concert with each other, kind of like more of a circular rather than just like a back and forth reaction. And these reactions can happen spontaneously. For instance, proteins can assemble or degrade over time, but they take hundreds of years to actually happen. And same with other chemical reactions. You can have these two products come together or get ripped apart, and it can happen spontaneously, but it doesn't have to happen at a time scale that we could utilize it. Could happen in years, it could happen in many years. And so enzymes are these proteins that specifically facilitate the what's called their catalyst, which speeds up the reaction. So enzymes work as a biological catalyst to speed up these reactions, to make them work on the scale of milliseconds rather than like years, for instance. And so these enzymes work to speed up these reactions by making the local environment around the product, the substrate, more favorable for that chemical reaction to happen. If you think about chemical reactions, they are affected by like the amount of water, the pH, the temperature, and things like that. And proteins can work to make those environments more favorable by having side chains that amino acids that have specific charges that can help change the local environment, or they can encapsulate your substrate and make it where there's very little water within it. And so that would change the aqueous environment and cause the chemical reaction speeds to change. And typically these enzymes are working to speed up these reactions, and they do so incredibly well. And so we utilize these enzymes in these metabolic pathways to generate high energy molecules in catabolic pathways. And then we use those high energy molecules to build larger molecules later on. And this is explained in this diagram here, where we take in nutrients or macromolecules of some kind and we break them down. And we break them down, and that breaking releases energy that we capture in the form of like electrons and things like that. And then those energy molecules can be used to build up molecules in anabolic pathways. And so we can essentially be recycling these macromolecules if any of these get damaged or old. And so enzymes are defined by as proteins that work to change these chemical reaction rates. They just speed them up. They don't create the reaction. Typically, you have a reaction, and there is like it would happen spontaneously, but you have this kind of resistance, and that resistance is called activation energy, and it's the amount of energy you have to put into the system before it can go into its products. And enzymes typically lower the needed activation energy. We'll have a picture of it in a minute. And so um, and then they also are reusable. So these enzymes can speed up multiple reactions over time. And so here's that activation energy. It's this hill of energy that is needed before substrates or reactants turn into products. And so enzymes lower that activation energy, and you can calculate this using like Gibbs free energy and things like that. And so when we talk about energy release and things, we're talking about this drop of energy potential. And we can capture that drop of energy potential to do some kind of work or to build up molecules in other pathways. And we'll talk about that in like glycolysis, Krebs cycle, and electron transport chain later. So we talked about this a little bit, that enzymes can be reused, but something that's important is that these enzymes are affecting the environment or manipulating the substrates. They can actually like rip apart like more mechanically, or they can put in electron donating groups that can help pinpoint to facilitate these reactions. But they're specialized to a particular substrate. They can't just like do that for every single reaction. They have to be tailored to a specific reaction. And they've co-evolved with these reactions and in these pathways to facilitate one particular function. And it does this by having that one particular place that substrate can bind. It's called an active site. And we can do whatever biochemistry or whatever method we use to facilitate lowering that activation energy. And excuse me. And let me go just to an active site real quick. And that substrate will bind into that active site and do some work. But because of the shape of that active site, because of how this protein is folded, not anything can go in there, just specific things can go in there. Okay. So now, enzymes don't have to just be proteins. So typically there are these strings of amino acids that are folded up into a complex shape. And not all of them are just amino acids. Sometimes they have a metal ion. Sometimes they have just chemical compounds associated with them. And sometimes they have other proteins associated with them. And so if you have non-protein material, you can call those enzymes conjugated. And those conjugated portions, those non-protein portions, can either be cofactors or coenzymes, where cofactors are non-organic and coenzymes are organic. And so these would be like if you like needed, for instance, some kind of, oh, I don't know, hormone that's just, or vitamin or something, a vitamin binding to the protein, that would be a coenzyme. Then you have cofactor, that would be something like a metal ion or something like that. And you can describe these enzymes as active or inactive, where when you have all of the parts that are supposed to be there, it'll probably be active. And if it's folded in the right way, it's going to be active. Or if you don't have all the parts, it can be inactive. And so when the active portion is called a holoenzyme, which has all of those components, it has the protein components and it has the conjugated cofactor. If you just have the protein and you don't have the cofactor, you can call this apoenzyme. You're not going to see these much more than in textbooks, but that's kind of how they talk about them. But these proteins are amino acid strings, and they are folded up into a particular shape, and that particular shape makes an active site to fit a particular substrate to facilitate a particular chemical reaction. And so everything is very organized and planned. And so how does it form that structure, and how does it form that structure reliably, right? And so there are four levels of structuring a protein. And the first one is called primary structure. And this is where you just look at the amino acids in the string of, the strand of amino acids, the polypeptide strand. You can read those amino acids, know what's next to each other, but you have no idea what that structure is going to be. That's called primary structure. This is where no interactions or intermolecular forces are happening on the strand. And then hydrogen bonding takes effect. So first off, primary structure, that's how it's synthesized, right? You're putting one amino acid on top of each other, connecting one onto the end of the other. And so you're making this strand. And so you're making this primary strand. As it comes off the ribosome, hydrogen bonds are going to take form and it's going to start forming secondary structure. And secondary structures are alpha helices and beta pleated sheets. Alpha helices are corkscrew shapes and beta pleated sheets are folded. You usually hear pleated as like folded fabric. So these secondary structures are formed using hydrogen bonds, and you'll have these two structures and then some strands of amino acids that are just in disordered loops that don't form these structures. And then you have tertiary structures where you have hydrogen bonds, van der Waals forces, disulfide bridges that bring together all of these secondary structures into a more tightly globulin form or a more tight fold. And this is where you're going to start getting interactions between your helixes or your beta pleated sheets and your disordered loops. They're all going to come together and fold into a 3D structure. And sometimes that's functional, but other times you're going to need multiple proteins to come together to make one functional protein to do some kind of job, right? And so that's called quaternary structure. And so here's an example of a primary structure where it's just that single strand. And we built this with an anabolic pathway, right? We're building in amino acids, and anabolic is building biomolecules. And then we have hydrogen bonds that form to form these secondary structures, alpha helices and beta pleated sheets, and these disordered loops. And then tertiary structure is where disulfide bridges, hydrogen bonds, van der Waals forces. Just a reminder, van der Waals forces are when you have two atoms and the electrons that move around in those atoms kind of, well, we'll just stay with one atom. You have one atom or one molecule, and you have electrons that move around it in some kind of probabilistic cloud or whatever. And sometimes they'll be on one side, all of them, or a lot of them. And they're not going to be equally distributed. When that happens, because electrons are negatively charged, you're going to have a temporary partial negative charge. And then you would have a partial positive charge, but it's temporary because those electrons are moving around. And so it happens. But when you look at it over time, it does exist for fractions of time. And those interactions or those partial charges can interact and hold things together weakly. And they're very weak. They're a weak intermolecular force. Then you have hydrogen bonds that are bondings between negative ions and hydrogen ions. So you have like oxygen and hydrogen that are binding together. Or like I think nitrogen fluoride. I'm not a chemist. I don't remember my chemistry. Don't quote me on that. I think it's oxygen, nitrogen, and fluorine that can create hydrogen bonds, but I would have to look that up to be completely sure. And then you have quaternary structure where you have those same intermolecular forces, but instead of inside the same protein, now you have two proteins that are interacting, and then there's a surface interface between them, and you have those same kind of interactions that are holding them together. Sometimes those active sites are in the middle of that joint, like right here, and sometimes the active site is not even close to that joint, but it's still important for the overall structure. You can have we talked about how specific enzymes are specifically tailored for specific substrates. You can describe this as lock and key mechanism. This is a very typical analogy of you have a protein that's a lock and you have a specific key, which is a substrate, and they can bind together and they only fit together to do this chemical reaction. But in truth, these proteins are strings of amino acids that are being held together by intermolecular forces. And so they're dynamic. There's some movement. These forces are holding themselves, these folds together, but there's still movement. And this is what leads to a more nuanced view of how proteins structure works. And this is called the induced fit model, where in truth, that active site might not be perfect, but it might be close. And so as it moves around, sometimes it won't fit because it's too small, for instance, and then sometimes it will fit because it's just right. And so this induced fit is more apt. And you can also have inhibitors that kind of block that induced fit too. We can talk about where these enzymes are located. So these proteins are folded and they do some jobs. Sometimes they're structural, sometimes they facilitate some chemical reaction. So you have structural proteins and then you have enzymes. These enzymes can be located inside the cell or outside of the cell. And if they're inside the cell, they're called endozymes. And you can think of endo as inside or entering the cell. While exozymes are outside of the cell, you're exiting the cell, exo or exit. Exoenzymes are secreted out into the environment to either digest nutrients, to communicate with other cells, to destroy the tissue or the environment to make it more favorable for yourself. So for instance, some bacteria, they'll release like keratinases and they'll try to get into the they'll break down the keratin of like your nail to get into it deeper, or you'll have collagenases, I think. Anyway, you have these secreted proteins and enzymes that can break down like the junctions between your cells. And so this can happen like on your skin to like get deeper into your tissue. This can happen in like your gut to get further into the gut lining. And those are called exoenzymes because they're secreted out, they're exiting the cell. And then enzymes can be regulated, or they can be really important and they need to be functional all of the time. So there's some chemical reactions that in metabolism you need to have going all of the time. You need to make ATP constantly because you need it all of the time to facilitate all of these reactions, these downstream reactions. You probably need to digest glucose, and you want to keep those proteins probably going. And so when you have these proteins that are constant at specific levels, these are called constitutive enzymes. These are typically required for life. And sometimes they're called housekeeping genes. So like in research, you'll use them as like a standard to be like, see, my samples have the same amount of protein because this enzyme is in the same concentration in all of my samples. Other times you have regulated enzymes, and these are enzymes that you have a when, where, and how. Sorry, you have a when and where you want to use them. And so, for instance, maybe in a specific time point in development is when you need that enzyme. Maybe you only want that enzyme when you notice food or when you detect a threat. And so you have mechanisms to turn on and turn off the expression of these enzymes. And these are called regulatory enzymes, and they are regulated typically at the genome level, sometimes on the protein level. You then have chemical reactions that are very typical that you'll see that are either anabolic, that build molecules up, and you have molecules that break it down, and these enzymes facilitate this. And a common one is a condensation reaction where we cool where you pull the water molecule from two molecules and you rip it off to make a covalent bond between two molecules. And this is a condensation because we're forming a water and putting it out into the environment. You can think of condensation as the formation of water on like a surface of a glass. That's water being condensated into the environment. In this case, we're joining two molecules together and putting water into the environment. And so it's condensation reaction. And because this is an anabolic reaction, because we're building molecules, there's some activation energy that's needed. And we use ATP, that high energy molecule that we use to kickstart our reactions to facilitate the breakage, the pulling up of this water and the formation of this covalent bond. The kind of the opposite of a condensation reaction is a hydrolysis reaction. And this is where we take water from our environment and we put it between two molecules and we essentially force them apart. And we do this, it's a catabolic reaction. We're breaking molecules apart. And so this is typically this doesn't require a large amount of activation energy. So we don't need a high energy molecule like ATP. However, there is some level of activation energy, but the enzyme is lowering that to a more manageable spot so that it happens more spontaneously. Lord, these are mouthfuls. Then we talked about how enzymes are like flexible and movable. They're relying on chemical bonds, hydrogen bonds. And hydrogen bonds are the attraction of hydrogens to negative charges, right? And so if you had a different pH, which is a concentration of hydrogen in a solution, the percent of hydrogen concentration, percent concentration of hydrogen ions at a log scale. But when you have low pHs, you have high concentrations of hydrogen ions. So maybe you have this negative charge, and sometimes it can bond to the hydrogen ion that makes the hydrogen bond that sustains that structure. Or other times, that negative bond could hydrogen bond with that lone hydrogen that's in solution. And so different pHs changes that equilibrium. And so it changes how those folds are held, and it can disrupt and disfold proteins. So if you wanted to denature your protein and unfold it, you can change the pH. You can add acid to it. You can do this with milk. You can put vinegar in your milk and it will curdle. And that's because the proteins are crashing out of solution because they are unfolding and then precipitate out. And so it's because we're adding those hydrogen ions and it's breaking those hydrogen bonds. Temperature does the same thing. We have things that are moving, and those hydrogen bonds are so long, they're only so big. And if you move things beyond that length, they're broken. And so temperature, when you have things moving at like a good amount of space because you have high temperature, high energy in a system, sometimes you can break those bonds and unfold your protein or denature it. And that's why there's only an optimum temperature that proteins can operate in, because at some point they're just going to misfold. And organisms that can withstand high pH, high, low pH, differences in pHs, or differences in temperature, they usually have specifically tailored proteins that can withstand that, or they have separate proteins that can help recover those damages as they occur. I'm in the weeds a little bit, so I apologize for that. But these enzymes change the speed of reactions, and you can also change the speed of that reaction using secondary molecules. And these are called inhibitors. You can also have enhancers, but we're talking about inhibitors. You can have competitive inhibition, and this is where kind of like that hydrogen bond could form where it could form structurally, or it could form with a free in solution hydrogen. We can have active sites that bind to the substrate that make our product, or we can just bind to some random willy-nilly thing that can fit inside of there. Maybe it doesn't even fit very well, so we can't facilitate any chemical reaction with it. But just by chance, because we're sometimes binding to the wrong thing, it's going to slow down our reaction speed. And so that's going to inhibit the reaction, slow it down. And because we're competing for the same active site, it's called competitive inhibition. Now there's non-competitive inhibition, and this is where something will bind to a different location, and this could change the shape of the active site. This could maybe bind just enough to block the active site, but it's not the active site, but it's binding right here. So now the substrate can't get into it. That's called non-competitive inhibition. When it is changing the shape of the active site, it's very specifically called allosteric inhibition, but allosteric inhibition is a type of non-competitive inhibition. Both of these non-competitive inhibition and allosteric inhibition are changing that speed, but it's doing so by not binding into the pocket, but somewhere else on that enzyme. And you can see this here, where competitive inhibition, you can bind to two different things, and one leads to that reaction and one doesn't. And so it lowers that reaction over term. You then have, or the speed. You can also have allosteric inhibition, where something binds to it and it can either change block the active site by changing the shape, or you can bind the substrate and it can make the reaction. Something that is mentioned, kind of depicted in this figure, but it's not always the case, is sometimes the products are the inhibitors. And why would you want to do this is if you're having a chemical reaction and you're just having it happen over and over and over and over again, you're going to pile up these products. You're going to accumulate them in high concentration to a point where you're not going to need them anymore, or they become toxic or whatever. So you need a system to be like, hold up, stop what you're doing. And so inhibition is a way to do that. And if we tie in the accumulation of our product to the inhibition of our enzyme, we have this negative feedback loop that stops it when we need to. And so sometimes the products will actually have a secondary binding site or an allosteric binding site to stop this reaction from happening. Now, this can also be something completely non-related. But just for the depiction of this neat mechanism, this does occur in the cells. You then have, we're going to now talk about glycolysis and how we actually break down sugars into energy molecules so that we can use them to do anabolic pathways in the cell. So typically, these energetic pathways can be described as consuming energy or releasing energy, depending on if it is releasing electrons or if it's releasing some kind of chemical energy that we can leverage, or if it's releasing it into the environment. You can, in chemistry, you see these as in just strictly as consuming energy or releasing energy. And so you can see a reaction of some product by some enzyme, facilitated by some enzyme, is creating these products and is releasing energy into the environment. So that's exergonic, while endergonic is absorbing that energy to make those products. And so usually these are, you're building things and you're needing to put energy in to build those things. Those activation energies are higher, and the energy states are usually higher. And so you need energy to go into that system. Well, with exergonic, you have an energy state of your products and your substrates. And so we're going kind of downhill from kind of talking about it in energy levels. And so usually that's releasing energy. Sometimes that energy can be leveraged and utilized to do downstream work. And we do that in glycolysis in the Krebs cycle and aerobic respiration, just to kind of incorporate the entirety. And so typically what we do is we're capturing that energy in high energy molecules like NADH, FDH, and ATP. ATP is the one that we're going to be trying to make the most of because it's kind of ubiquitous within the cell. It's used for all kinds of reactions, and it's usually considered the currency of the cell, and it used to kickstart reactions by lowering that activation energy or providing the energy to overcome that activation energy. And so we make these high energy molecules through redox reactions where we can transfer electrons from one molecule to another. And so there's something called reduction and oxidation. And the one way you can remember this is oil rig, where oxidation is loss and reduction is gained. That's how I remember it, at least. And so when you have a molecule that loses electrons, it's oxidized. And when you have electrons that are gained, they're reduced. And so you can remember the acronym oil rig. Oxidation is loss and reduction is gained. So now let me try to get rid of those markings and get my pointer back. We'll just exit and come back because I don't know how to work PowerPoint. So you can look at glycolysis, where we break down glucose into component carbon dioxide and water as a redox reaction, where we have the sugars that are being oxidized into CO2 and the oxygen that is being reduced into water by remember, reduction is gained. So these water molecules, these oxygens, gained electrons. So you can think of the process of breaking down glucose as a energetically downhill process of we're breaking down this glucose. Remember, catabolic is breaking down, and we're doing so in a oxidative way. So we're going to be losing electrons, and we're going to harvest those electrons onto high energy carrying molecules that we can use for downstream biochemistry pathways. And so we make ATP at the very end of this, which is the currency within the cell that's a high energy carrying molecule. It usually binds to proteins quite often, and so then it breaks these terminal phosphate bonds, and it breaks this triphosphate sorry, I have functional movement disorder, so my brain sometimes breaks. So what this is you have a adenosine ribose sugar, which is like the kind of the building block that start in one phosphate at least. That's the build, which is the backbone of RNA. And so there's a lot of these molecules in the cell, but there's a lot of phosphate. Excuse me. So sorry. And so there's a lot of this base molecule, this backbone molecule, but we can put these inorganic phosphates onto it, and each one of these holds more and more energy within the bonds, or at least they can be used to facilitate more energy in the bonds. And so when we break this terminal phosphate bond, it releases this inorganic phosphate, and we can put that inorganic phosphate onto another molecule. And that process of transferring, it's not really the breaking of the bond, but that's the transferring of this inorganic phosphate onto the protein or onto the substrate itself, is enough to overcome the activation energy. And so we have a ton of this molecule, and it facilitates a ton of reactions. I know I'm going in circles. That's the functional movement disorder, and I do apologize about that. But ATP can be formed in a couple of different ways. We can physically rip off the phosphate and put it on to ADP, which is when we only have two. So I guess I'll say this again. The backbone, when you just have one phosphate, is called adenosine monophosphate, mono meaning one. Diphosphate would be when there's two, and adenosine triphosphate is when there's three. So ATP is the high energy carrying molecule. ADP is when that phosphate has been released, and AMP is what's used to build RNA. And so you can take a phosphate and put it directly onto ADP to make ATP. And this is called substrate level phosphorylation. You also have oxidative phosphorylation, where we use high energy carrying molecules to make a proton gradient that we then use to drive a motor to just essentially put these phosphates onto these ADP at high fidelity or a high rate. You can also do this sunlight, and it is called photophosphorylation. And so we can try to break down sugars into energy in a couple of different ways. And in truth, you have countless ways. Each organism might have slightly different enzymes, and so they might have slightly different pathways, or their environment facilitates different pathways. But for the most part, aerobic respiration, anaerobic respiration, and fermentation generally are the three main types of breaking down that catabolic pathway of breaking down sugars and macromolecules into and harvesting energy from those macromolecules. And so we have three steps in those processes of breaking down sugars of glycolysis, Krebs cycle, and respiratory chain, where each of these aerobic, anaerobic, and fermentation kind of have slight variations of them. So you have first glycolysis, and this is where we first start with glucose. And glucose is broken down into two pyruvate molecules. And that is the whole of glycolysis. It's more complicated than that, but that is the starting and the end products of glycolysis. We have a couple of high energy molecules that we've generated in the process. And these high energy molecules we can utilize to do some work later on in this process downstream. Then we have the Krebs cycle, which takes this pyruvate, makes it into acetyl-CoA. We'll talk about that in a minute. And it goes through a cycle. And it keeps breaking down these pyruvates into component carbon dioxide by ripping off carbons slowly and generating high energy molecules. And those high energy molecules, again, are going to be utilized the electron transport chain downstream. So we have aerobic respiration. Aerobic is with oxygen. And you have anaerobic, which is without oxygen. And the AN is the without prefix. And so when you look at this word, you have aerobic, which is the oxygen part. And then the AN is the without, or it negates this root. And so it's without oxygen. And so oxygen is actually used at the very end of this process to kind of cap, and you are used as the final electron acceptor. That's a very important word to remember. In aerobic respiration, oxygen acts as the final electron acceptor. And this is what caps and stops this reaction. And it makes it a little bit less damaging to the cells. Without oxygen, it would be damaging. Because you would have free oxygen radicals and electrons that aren't bound to anything, and they could just bind to anything willy-nilly and maybe wreak some havoc there. Anaerobic respiration, because you don't have that oxygen to cap the reaction for that final electron acceptor, what are you going to do? You're without oxygen. So what they do is they use in non-organic substances like sulfates, nitrates, and they or nitrites, and those are what they use to accept those electrons. Electrons can bind to these and cap that reaction that way. And fermentation, on the other hand, uses organic molecules, which again have carbon and hydrogen backbones to catch those electrons that are formed throughout this process. And so the biggest thing about aerobic, anaerobic, and fermentation is the final electron acceptor is what's mainly different. In fermentation, you're not going through the Krebs cycle and electron transport chain, so that is pretty notable. And so typically fermentation is the breakdown of sugars into acids, gases, and alcohols. And so you can think of it that way. And we'll see that in lab. I keep going back to the slide. You'll see this in lab as we'll see changes of pH with the production of acids. We'll see bubble formation with the production of gases. And we don't actually look at alcohols much, so we won't see that in lab, but you could look at that if you wanted. And so what's notable also is that this breakdown of glucose can generate a high level of ATPs. So we can generate 38 ATPs under ideal circumstances. Because it's biology, it's a little bit less than that because it's always an inefficient process. But the reason this is occurring is because glucose has a lot of high energy, is in a high energy state. And we can rip it off to molecules that have lower energy states or have lower energy bonds, like carbon dioxide. And so as we generate carbon dioxide, which is what we expel when we breathe, it's from this process, we can take advantage of that drop in energy potential of these molecules to drive the process. And we do that by taking off the electrons and capturing them into high energy bonds. So glucose is a catabolic reaction, breaking up of a molecule. Catabolic pathways are breaking down of molecules, and anabolic is building it up, but glycolysis is breaking down of a glucose. The biggest thing you're wanting to know is glucose is what starts this reaction. And then the first step is glucose 6-phosphate. So we take an inorganic phosphate and just stick it onto the glucose. This is an important first step because it puts a charged molecule. That phosphate is a polar molecule, and so because of that polarity, it locks the glucose inside the cell. So we talked about how the membrane, non-polar molecules without a charge can move in and out freely through diffusion, but charged particles, they don't move through the membrane because of the hydrophobic tails of the phospholipid bilayer. And so because the phosphate is charged, it can't go through that membrane through diffusion. And so when we put that phosphate onto glucose, we are essentially locking it into the cell and committing it to this pathway. We can then rearrange the glucose to fructose 6-phosphate. You don't need to know all of these steps or and the names of each of these things, but you kind of need to understand what's happening, that we're taking glucose, locking it in a cell through phosphorylation, then we rearrange it, and we slowly are going to be breaking it up into 2 molecules of pyruvic acid. And in that process, we have to donate and break some ATP to kind of get over that activation energy. And so we're investing some energy, and we break up this molecule into 2 molecules that are similar, but they can rearrange sterically into the same molecule. Excuse me. And then we can process this, and we can start actually capturing energy from this molecule by making 2 high energy NADH plus molecules, so that we're pulling off hydrogen ions. And NAD plus has an open charge. We take that electron and we make NADH, and we this is going to hold that those hydrogen ions and those electrons to the electron transport chain. And these are called high energy carrying molecules. And that's why they're called carrying molecules, because they're going from this place of glycolysis to the respiratory train, or the electron transport chain, depending on this, what you call it. So then you have ATP starting to be you have phosphate groups that are getting ripped off of the substrate of this 1,3,6 bis phosphoglyceric acid, I'm having a horrible time. But this molecule has phosphate. We can rip it off and we put it onto adenosine diphosphate. And this makes adenosine triphosphate, which is a substrate level phosphorylation, where we're taking the phosphate and putting it directly onto the ADP using an enzyme. We do this again all the way through some rearrangements until we get into pyruvic acid. So again, you have glucose that's coming in. We're putting in some energy at this energy investment phase, and then we're harvesting some energy at the energy harvesting phase to generate 2 pyruvic acids. And those 2 pyruvic acids come from these molecules that were separated. And then we have 2 of everything that's from this point on. Overall, this is kind of what you need to know, where glucose is being turned into pyruvic acid and it's generating high energy NADH molecules and ATP, and it generates 2 ATP. And this is net ATP. If you remember, we used an ATP and then we got 2 ATP. And so there's a net of 2 ATP positive. So eventually this pyruvic acid is going to go into the Krebs cycle. And before it goes into the Krebs cycle, it first needs to be conjugated with coenzyme A, which is just this organic molecule that helps facilitate enzymatic function. Remember, coenzyme is a cofactor that binds to an enzyme to make it functional. And so this coenzyme A is going to bind to pyruvic acid to make acetyl-CoA. And acetyl-CoA is going to go into the Krebs cycle by first binding with the end product of the cycle, which is oxalic acetate. And this is what makes it a cycle, right? So the end product gets joins with this incoming new stuff. And then we can start this process all over, because when you join those together, it creates citrate. And citrate is the first molecule in this chain. Through a rearrangement, it creates isocitrate. We can harvest some of these hydrogen ions and pull off some electrons to make a high energy molecule. And we also rip off a carbon. And so this creates CO2. And so this process creates CO2. And so this is where we're exhaling all that carbon dioxide gas from respiration. Or the bacteria are exhaling this in aerobic respiration or anaerobic respiration, either way. So then isocitrate is then, after a carbon is removed in those hydrogens, it makes alpha ketoglutarate, which then turns into succinate and CoA and succinate and fumarate. These are just either different arrangements or conjugations of different molecules that are being attached to it to kind of help facilitate this process. It kind of manipulates this energetic barrier so that we can essentially rip off one of these carbon dioxides and some of the hydrogens to make these high energy carrying molecules. Again, you're not going to need to know these steps. You're not going to need to know the structures of these things, but you're going to need to understand that the process of this is we're essentially breaking down the carbons off of it, and in the process pulling off hydrogens for high carrying energy molecules, and also expelling carbon dioxide. And then eventually we get to malate and then back to oxalic acetate, which is going to then bind back with oxalic acetyl-CoA back into citrate, and so on and so forth. And so this is like the generalized acetyl-CoA comes in and it generates these high energy carrying molecules and expels carbon dioxide, right? And so these high energy carrying molecules, we've been generating so many of them. What are we going to do with them? And it's called electron transport chain or the respiratory chain, depending on like what organism that you're dealing with. But like they're essentially the same thing, kind of like glossing over some nuances, probably, right? So it's essentially a series of proteins that are embedded in a membrane that shuttle electrons and protons through a membrane to facilitate a gradient, a chemical gradient that we can utilize to do work. And we'll show that here. So we have in eukaryotes, you have the mitochondria, but in prokaryotes, you have the intermembrane space, the periplasmic intermembrane space, right? The periplasmic space. The mitochondria is called the intermembrane space. Technicalities. So it's important distinctions, but it's hard to keep them all straight sometimes. So you have these high energy carrying molecules that have these hydrogen ions, which are protons, and you have electrons, right? Hydrogen is one proton and one electron. They bind together and make the hydrogen atom. And so we essentially pass the electrons through a series of proteins, and this literally these electrons are being transferred through this system, and this facilitates the pumping of the protons into this space. And so this is allowing us to kind of separate the positive charge from the negative charge. We pump the negative charge back into in this case, it's the mitochondria. In bacteria, it's inside the cytosol, inside the cell. And this is where oxygen comes in, because this is where these electrons, those could wreak havoc. But we also have oxygen radicals that bind to them and form water. We have oxygen that's ripped apart and it takes on these electrons and protons to make water. And this is the final electron acceptor. It's taking the electrons from this chain, right? The electron transport chain, and the oxygen is accepting it and forms water. And so again, when you exhale, you'll have some moisture, and that is the water that is being expelled from a respiration. So we have this chemical gradient, but why are we doing it, right? So you have a positive charge and a negative charge. There's a gradient of charge. We can utilize that to do work, right? It's like a battery. It's not a battery, but it's like a battery, right? And so you have what's called ATP synthase, which takes advantage of this chemical gradient. And we can utilize the chemical gradient to do this work, to move the protein, to take an ADP and a phosphate and put them together to make an ATP. And this is a rotary motor, so the movement of it is actually like twisting, and as these protons move through it, it causes the twisting to occur. And so the something that's basic, kind of like fundamental like concepts is diffusion. And so things want to be at equal concentrations across the solution. And so if you have high concentrations in one area, those high concentrations are going to move and diffuse into lower concentrations till to make it equal. And so we're going to be using what's called chemoosmosis, or we're going to be naming this gradient, or this desire for these ions to kind of be equalized, as the proton motive force, right? The protons are providing like a movement or a motion. And so that's kind of where the name comes from. And so these ions want to get away from this high concentration into this low concentration. So they really want to move through this process, and the ATP synthase facilitates that process. And so as these protons move through, it brings together the phosphate and the ADP into high energy carrying molecules. And this can generate like 30 or so ATP, or 28, like high numbers of ATP. And so this is where a lot of our ATP is generated, even though glycolysis did generate two and Krebs cycle did generate some, this is where that meat is. And in bacteria, this is, we don't have organ bound, we don't have membrane bound organelles. And so there's not very many places that we could create this chemical gradient. And so they take advantage of this like pretty much the only one of the few compartments within the bacteria cell is this periplasmic space, which is between the cell membrane and the cell wall, which is made of peptidoglycan. And they essentially pump the electrons through the same kind of protein chain, and they create this gradient of protons that then can be used through ATP synthase to generate ATP. So let's try to get this back to a normal size. Perfect. Oh, they already had one zoomed in. Forgot about that. And so this like diffusion, or this desire of to equalize the charge, is called chemoosmosis. And again, we call it the protomotive force. Chemoosmosis is like the movement of chemicals across a gradient of a concentration gradient. Osmosis in general would be like the movement of water. So chemoosmosis is the movement of chemicals across that concentration gradient, while osmosis is just the movement of water across some chemical gradient. Because water is wanting to also facilitate that change. So we talked about the oxygen is that final electron acceptor from the electron transport chain, or the electron transport system. The electron transport chain is specifically to eukaryotes, and so the electron transport system is more precise to prokaryotes, if I remember right. Again, glucose is made into glucose 6-phosphate, broken down into 2 pyruvates, generating a couple of ATP in high energy carrying molecules like NADH. Then through the Krebs cycle, we generate more NADH, ATP, and FADH2. And then we can take those high energy carrying molecules into the electron transport system and make this concentration gradient of this proton gradient to drive ATP synthase to make ATP. And so anaerobic respiration, instead of using oxygen at that final electron, except uses nitrate, nitrite, and like sulfate to facilitate those the binding of those electrons as they come off of that electron transport system. And then you have fermentation, where they kind of lack that Krebs cycle, and they lack that electron transport system. And so they rely on the breaking down of glucose into acids, gases, and alcohols to generate those high energy carrying molecules, or ATP. And so they have vast variety of biochemical pathways that can do break down different things, and each species have different enzymes, and those different enzymes can do different things. So lactobacillus can make lactic acid, while yeast make ethanol, for instance. There can be some overlap in these, but not everything makes all of it, for instance. Another thing to mention about the intermediates of glycolysis and the Krebs cycle is that those like alpha ketoglutarate, succinate, citrate, all of the pyruvate, all of those things have other things that they could be turned into other than this. And so, yes, that's being broken down into energy, but it can also be turned into signaling molecules, hormones, other amino acids. That's a really big one. Alpha ketoglutarate, I'm pretty sure, turns into a lot of amino acids. And so this process is like a core foundation to so many other side reactions and biochemistry that facilitate and sustain your cells. So it's really important to remember that these things aren't just independent from one another, but they're very connected. So like, for instance, like the synthesis of amino acids that are really important for proteins. And so without amino acids, without being able to break down glucose and making those amino acids, you're not going to be able to make your proteins and enzymes, for instance. Very well. And so with that, that is chapter 9. Sorry that I had some functional issues today and that it seems like the lecture took a long time too. So I apologize about that. I hope you had a great time. Bye-bye. Oh, hi.